My pleasure to introduce Professor Ahmed Mustafa. He's a professor of clinical pathology. He's a clinical director of the HLA lab, Istanbul Hospital. He's assistant professor, Department of Pathology and Lab Medicine, University of Saskatchewan, Canada. He will address today basic immunology and nomenclatures. Fadal.
Thank you so much for this nice introduction. And actually, it's a pleasure for me to be in the middle of the nice Ain Shams group, although I was a Cairo University graduate. So this was it's actually a good pleasure to me. And thank Dr. Magdi for the kind invitation. It's actually it's an honor to me to come here. So I'm actually going to switch gear, and I'm going to talk more about one of the very silly subject. I didn't like it when I was an undergraduate, so we'll see what we can say here. So this slide is not intentional for my professor and senior. This was more for the junior colleagues we have here. So what does HLA stand for? Is it high-level assessment? Is it high-layer compatibility? Is it heated living area? Is it human locus A? Or it's human leukocyte antigen? So most of you, I'm pretty sure, know that it's actually human leukocyte antigen. But also answer number D is correct, because this was the original name in 1960 when they originally discovered it, before they know now that we have almost 30,000 alleles of HLA. So human comes from human, and leukocyte, because all the original experiment which was done on the white blood cells. Later on, they found that human leukocyte antigen is expressed in all nucleated cells. So if I ask you, red blood cells, does it express? No, because red blood cells does not have a nucleus. But if I ask you, platelets does express, although it does not have a nucleus, but it actually originates from the megakaryocytes, and then it becomes fragmented. And platelets is a major sensitizing event. You can get a PRA or CPRA of 95% from a couple units of platelets. And antigen, because it can elicit an immune response in the form of antibodies. So if I ask you about this picture here, are this image of the same person? I'll say some yeah. So some people will say, yeah, they are the same person. Some will say, no, this is Ibrahim, this is Hossein, and some will have no clue. But in order to know which one is which, you can do a fingerprints. Although you are identical twins, your fingerprints will be different. So if we did a fingerprints here and the fingerprints are the same, then they are the same person. But if we did a fingerprints and they are different, then they are actually two identical siblings. And the same scenario for HLA. So if I ask you, does this white blood cells belong to this person? The answer will be, yeah, they look similar, but we need more information. And the information will be guided through the HLA. So now if I ask you, look at both HLA, are they similar? Yeah, they look similar. Both of them are white, so yeah, they look similar. So we call them HLA identical, or in our language, we call them matched HLA. But if we look at the other case here, one HLA has different colors, and the other one is white. So now their HLA is not identical or mismatched HLA. And actually the field of transplantation started in early 1941 by this Nobel Prize winner George Snell, when he did some experiment on mice. So basically what he did, he transplanted a skin graft from one inbred mice to the other, and it was accepted because they were in the same kind. But when he used a different kind of mice, their graft was rejected. So he found that this was very interesting. So he started to play some games. So he get a mice which is A and mated to another mice B, And then now the offspring is A and B, so they carry the genes from both parents. So when they transplanted the skin from the parent to the offspring, the graft was accepted, but it was rejected on the other way when they put it back from the parents, from the offspring back to the parents, because now the parents will look at the B as a mismatched antigen and it will form rejection. However, in the field of human, the field started by another Nobel Prize winner, John Doucet, who was one time bored in his lab. So he asked the nurse to bring him some blood, and he wanted to do some games during in his lab. So he actually isolated the white blood cells. And at that time, they didn't know that the white blood cells actually express HLA. And he put one drop of white blood cells on a slide, and then he asked the nurse to bring him plasma from people who has been previously transfused. So now we know that people who has previously transfused, they probably have a lot of HLA antibodies, but they didn't know at this time. So he took the serum, and yeah, we didn't know that they have antibody, and then they put a drop of the serum on the top of the white blood cells. And he started to examine the reaction by the microscope. The interesting part here, he found sometimes the serum agglutinates the white blood cells, and sometimes it does not agglutinate. But what he found interesting is that the patient himself does not agglutinate his own white blood cells. And this was the discovery of the HLA. He did a lot of experiment, and the first gene that was discovered was called MAC, M-A-C, which now we know as the HLA-A2. So the structure of HLA is, we have two major classes here. So we have the class 1, which is formed of a long alpha polypeptide chain and the beta-2 microglobulin. So if we did typing for the beta-2 microglobulin for all of us, it will be the same. But the alpha chain is one of the highest polymorphic region, and there is almost 20,000 HLA class 1. The most common class 1 that we test for is the HLA-A, the HLA-B, and the HLA-C. Now, if you look at the class 2 is formed of an alpha and a beta chain. So alpha and the beta chain is a little bit different. So for the DR, we don't do the typing for the alpha chain, but for the DQ and the DP, we do the typing for them because they are polymorphic. And the most common one that we do typing are the HLA-DR, the HLA-DQ, and the HLA-DP. So anything which starts with a D is considered class 2. So now let's see how the normal function of the HLA. So HLA is actually, normal function is not to do anything with transplant. The normal function of the HLA is just to present foreign peptides to the T cells to let the T cell there is something dangerous inside the cell. So if in order to understand what happened after transplant, let's look at the first example here. This is normal individual. The normal individual have a self-kidney cell, and the self-kidney cell will have self-HLA expressed on the surface. So I put them here red, so you know that red and red, they are self. And you can see there is a small blue dot. This is actually a self-peptide. Sometimes there is organelles inside the cells that get degraded, and then they are expressed on the HLA. But again, the T cells want to make sure that this is actually a cell. So it comes and screens the kidney cells, and if it didn't find anything, it will just leave it. But if we go into the transplanted organ, now you can see the kidney cell have a different HLA class 1 than the self. So there are two different kinds of recognition. There is a direct recognition and the indirect recognition. And the direct one is actually happening very, very early in transplant. And this is because when you transplant the kidney have some leftover donor cells inside it. So once the kidney is flushed with the recipient, some of these donor cells escape, And now this donor cells is the lymphocyte, probably, and they have the foreign HLA. So now when the T cells see them, the T cells will stick to them. They will not leave them, because now the T cell recognize that this is actually a foreign antigen. And this is in case of direct allorecognition, because it's a direct relation. Now, sometimes you can get this HLA shed out of the cell, and now they are going to be taken by the scavengers. And then, because they are foreign, they will be presented on the self. Now see, this is a self HLA. They are going to be presented. So the T cells will come and see them, and the T cells will recognize, yeah, they are self antigen, but they carry non-self protein. And then the T cells will become activated. It will go to the lymph node, and now it become an effector T cells and come back and destroy the kidney. So there are, as I mentioned, HLA is the most polymorphic region in our system. Here I just put a snapshot of the commonest serological or recognized antigen that we know. So for example, we have like 28 recognized antigen for A, 50 for B, and so on here. And then every time they discover one antigen, it got a number. So you can see the first one they discovered was called A1, the next one was A2. And the system is a little bit complicated, because you can see it's jumped from A3 to A9, because the one that they found from A4 to the 8, it was wrong, but they already gave the number to 9, so they will not go back and rename them. Otherwise, it will be like a vicious circle. And here is actually now what we can see in this one is like 2022, and this is 1987. So, and this is the number of HLA alleles they discovered. So you can see that we have almost 24,000 HLA class 1 genes and 8 or 9,000 of the class 2. And it actually was related to the method of typing that they were using. So in early stages, it was the CDC that was the gold standard for HLA typing. And then when molecular became available, they discovered more alleles. And then now when next generation sequence are available, they discover massive amount of alleles. And if they go to a different method, they will find more and more alleles. And this, it's always evolving. So, because all these 30,000 HLA alleles that we have, the HLA nomenclature committee, which is part of the WHO, has to decide on how to name this HLA. So they put a system, because if you keep going from A1, A2, A26,000, this is going to be a lot of work. So they decided to make it much simpler. So in order to put it, you have to put the word HLA hyphen and then the name of the first gene. So if it's A, you put A. If it's B, you put B. If it's DR, you put DR, followed by an asterisk, and then followed by the first number. This is the serological equivalent. And then when they started to do molecular, they put now the molecular typing. So this is just like could be a single or a single mutation that happened to the main one that is not sufficient to change the complete protein structure, but is sufficient to change the SNP. And this one, if when they started to see some of the mutation does not affect the protein structure, so they give it another number. It's not very clinically significant. And when I say it's not clinically significant until today, I'm not sure what's happening in the 5 years, because I know now people are recording my lectures. Someone will match it in the future. And again, this is also not clinically significant. This is what they look at the intron levels. If there is a mutation at the intron, not the exons, they give them the number. And again, there are a lot of research now finding that these are significant, but it's not clinically significant until today. So let's see much more a practical example and what we do in our lab. So for example, here you can see that this is a kidney recipient HLA typing, and it has a donor 1 HLA typing. And in the lab, what we do, we start to compare. It's like a puzzle. So we compare A1 to A1, yeah, they are matched. B8 to B8, they are matched, and so on. So now, this is a case of an HLA identical match between the patient and the recipient, and we call them zero antigen mismatch because there is no antigen mismatch between the donor and the patient. But if we look here at a different example, and you can compare the A1 to the A2, it's a mismatched. You compare the B7 to the B8, it's a mismatch, and so on here. You can find that the patient and the donor too is complete mismatch. So now they are mismatched at the 6 antigen. So we call them 6 antigen mismatch. But unfortunately, we cannot match every patient to every donor. Otherwise, no one will get transplanted because the chances to find an identical HLA matched donor is 1 over 20,000. And you can wait on the wait list for 20,000 years until you can get it. But what we do in the lab, we do what we call compatible match. So we want to make sure that the patient does not have antibodies against the donor. So for example here, if this patient has an antibody against the A1 or an antibody against the DR7, now this is not a compatible donor. And we have to look for a different donor. But I don't want someone to come and say, oh, the HLA match does not matter. No, it matters. And this actually, some researchers has done over and over the years. And they found that if you have a zero mismatch, they have the highest survival rate. If you have the 6 mismatch, you have the lowest survival rate. And someone may say, oh, yeah, but maybe in the early 80s, they were not taking tacrolimus. No, even when you are taking tacrolimus, this is the case as well. And something, a very important feature about the HLA, it's actually inherited as one set of alleles or one set of haplotype. So for example, here you can see that these 2 parents, the parent has the blue and the green, and the mother has the, I don't know, orange and purple. So when they get offspring, you can predict the typing of their offspring because it's actually inherited as one set of haplotype. So maybe children are like the first child can get one haplotype from the dad, and then the brown from the mother, and the other one, and the other one, and the other one, and the other one. So what is the significance of that? The significance, if you have a patient and come with parents, you can tell them your parent will be a good match to you because they actually carry 50% of their gene. The other important thing here is that if the siblings come together, the chances to have an identical matched HLA sibling is 25%. And HLA is actually codominantly expressed. So it's not like the blood group. Blood group, usually the dominant is expressed and the recessive is not. But the HLA is actually, both of them are expressed. And as I mentioned, the function of HLA was not for transplant, but the function is to present different peptide to the immune system. So it actually need to be diverse. So you can see here, the A1, for example, comes from the dad. The A2 comes from the mom. The B27 comes from the dad. The B8 comes from the mom. And this is, I'm enough now with all this boring stuff, and let's go to more clinical stuff. And this is how our report looks like. This is the report that we do in our lab, and we give it to the transplant nephrologist to see. So first, we put the blood group. Although we don't do the blood group in our lab, it's done in the transfusion medicine. But because this is the first factor, the transplant nephrologists like to see they are compatible blood group. And then here, what you see is the class 1, and you can see the A, B, C, and we follow the nomenclature committee in identifying. You can see that there is a small asterisk. And this is the class 2, and again, this is the DR, the DQ, and the DP. And this is actually a high resolution typing. So how does our system recognize that the kidney is foreign? So as I mentioned before, HLA is expressed on all nucleated cells. So here you can have HLA is, oh, I'm running out of time. So you can have, this one is expressing HLA, so if a donor is formed against the HLA, it will bind to it. This will attract more complement cascades, and then this will result in destruction of the kidney. In the lab, what we do, we measure the donor-specific antibody, and we take a biopsy, and we look at the C4D. And this is how we diagnose AMR, plus the pathological findings. There are 3 different kinds of AMR. The hyperacute. This, if we see hyperacute, this will be a chain to the HLA lab. That means that we did not do a good job, because we have to screen the patient very well before transplant. But you can see it. The only time I have seen it is when we did an incompatible blood transfusion, and in the spot, the kidney was rejected. But this was a special case. I can talk about it later. The acute, this actually happened between week and year, and this is because of the formation of new HLA antibody. And chronic is much more common, is when the people become not adherents to their therapy, and they're starting to lose the graft. And again, this is not what we want to see. Oh, so should I stop? So I'm going to talk now about HLA antibodies. And the most common sensitizing event, if someone already been transplanted before, or if it's because now we have foreign antigen inside their body, or because of pregnancy, because now the mother are carrying half of the genes from the dad, and blood transfusion, and I already mentioned that, and sometimes vaccination. And I have a paper just got published last week. We found that COVID-19 vaccination can result in a lot of formation of HLA antibodies. It's published in Archive of Pathology if you are interested in reading it. And now that I'm going to talk about 3 different definitions. We have what we call the calculated PRA, or the CPRA, the current CPRA, and the cumulative CPRA. So first, what is the calculated panel reactive antibody? So CPRA predict the percentage of donor in a specific population that is likely going to be incompatible with a given recipient. So for example, the frequency of HLA-A2 is the most common in Caucasians. So North American and European, most of them have A2. And the frequency of this gene is 40%. So 4 out of these individuals will have A2. Now, if we have a patient who has an HLA-A2 antibodies, this patient will react with 40% of this individual, and there will be a virtual cross-match positive. So the only chance he has is only to get donors from 60%. So his CPRA, or the calculated panel reactive antibody, is 40%. It's very important when we go to this deceased donor transplant, because we need to know the likelihood that they're going to find a suitable donor. Cumulative CPRA or versus current CPRA. Cumulative, that means that every time the patient is on the wait list, new antibodies are formed, and they are cumulative. We don't delete them. They are historical. They keep in the system. They are important when we do the virtual cross-match, because when we do a virtual cross-match, we look at the entire history of the patient over the entire waiting list. So for example here, if you have someone who had an antibody detected in 2002, but in 2003 the patient did not have any antibody, then his current CPRA will be 0%, but his cumulative is 40% because he accumulated over the year. And this is an example of a virtual, you can see this is the current serum, has 0 PRA, but we put also the cumulative because he had some previous historical HLA. So if I'm going to talk about a virtual cross-match, it's used to predict the result of the physical cross-match. So what we need to do the virtual cross-match, we need the HLA typing of the patient and the HLA antibodies, and we need the donor HLA typing, and we start to compare between them. So if we have a waitlist patient, every 3 months this waitlist patient put serum, and we test for the antibodies, and we put the results in the computer system. And then now if you have a donor who came, the donor will be HLA typed, and the donor typing will be added to the same computer system. The computer will start to look at the patient typing, the patient antibodies, and compare it to the to the donor HLA typing, and then it will create a virtual cross-match. It will shortlist this patient. And this is just an example of a virtual cross-match report. You can see here is the donor, and these are compatible to this donor because they don't have antibodies against the donor. I can skip this, and then the physical cross-match is the same as the virtual, but we do it physically. So basically, we incubate the patient's serum with the donor cells. So here you can have the patient has multiple antibodies, and if we incubate them and the patient has the antigen that is binding to this antibody, they will bind. We add another antibody which has a fluorescence in it, and if it binds, then means that this will result in a positive cross match. I think at the end I would like to thank you for listening to me, and sorry for taking longer than my expected time.
Many thanks, Professor Ahmed Mustafa, for your illustrative lectures.